Hi, everyone, and welcome back to the last session of the conference for day one. We're going to start this final session with the UFAW Awards presentation. So over to you about the awards.
Thanks, Lisa. I'm really delighted, as every year, to be able to present UFAW's awards, which we give every year to scientists who've had a really outstanding achievement in animal welfare science. So we give awards for early career researcher of the year to people who are early on in their career but have started off making a really impressive impact even in the first few years of their academic career. And we also give a medal for outstanding contribution to animal welfare science. So I've already told you who the winners are, but just to reiterate, our early career researchers of the year this year, we have two award winners. So first of all, I'm delighted to announce Dr. Sarah Hintze from Vienna, who is an award winner for her research, which is predominantly focused on trying to understand something we've been talking a lot about today, which is the affective state of individual animals. She's done lots of work on really difficult methods, like trying to use judgment bias to understand the affective state of individual animals, and also more recently been looking at boredom in pigs, which again is probably not the easiest thing to try and measure, but incredibly important for us to understand what's actually going on for those individual animals. She was nominated by her colleagues and peers who also praised her public outreach and supervision work. And please do note that she'll be talking about her work at 2:20 tomorrow, so you'll be able to hear much more about her work. So I won't say too much more about the work today, but I think Sarah is here, so she can say a couple of words if she'd like to, and I'd like to offer her enormous congratulations from UFAW on being one of our early career researchers of the year. So Sarah, if you're out there.
I am. Thank you very, very much. I feel very honored to receive this award, and I would like to express a very huge thank you to UFAW for getting it. Of course, as is usually the case, I would not be here today without the support of very many people around me. And first of all, as you have said already, I was nominated for this award, and my nomination got a lot of support by many people, and I would like to thank you for this. I'm in the very lucky situation that I have experienced so far the wonderful combination of freedom to do whatever I wanted and still want to do, but on the other side also being incredibly supported, first by my PhD supervisor, Hanno Wirbel in Switzerland, and since then by my postdoctoral mentor, Christoph Winkler. And Christoph, Hanno, I really owe you both a lot. Thank you. My luck did not end with wonderful supervisors, but during the past years, I met so many inspiring and creative students and other researchers, many of them being close friends today. Already during my PhD, I was challenged, inspired, and supported by great colleagues and friends in Bern in Switzerland. For example, just brainstorming what our ideal research center for studying animal emotions would look like was already a very joyful experience. Moving on to Vienna, I again found very great sparring partners, both here at BOKU, but also at the VetMed University. And these valuable connections have since then been complemented by researchers from all over the world, and I'm very thankful to be part of this international animal welfare research family. So I would like to thank all of you a lot. Thank you, UFAW, for awarding me this prize. Thank you.
Thank you very much, and congratulations again. The only shame with these online conferences is that you can't hear the applause in a real room, but there'll be a certificate on the way to you as well soon. So congratulations again, and thank you very much. Of course, as happens in so many years, we had lots of really great nominations for early career researcher of the year, and we couldn't split them. So we actually have a joint award this year, and the other winner of the award is Dr. Jordan Hampton from Australia, who works predominantly on the welfare of wild animals. He does lots of practical animal welfare science concerning the welfare of wild animals, including Australian native species. But he also was nominated by his colleagues for the work he does at a different level, thinking about the ethics of the way we interact with animals and manage wild animals in particular. And as I mentioned earlier, he's a co-author on a really important book on the ethics of wildlife management, which UFAW will be publishing later this year. So a completely different field and a different approach, but equally praiseworthy and really impressive career for someone so early in their academic career. I think Jordan is joining us by video because it's probably the middle of the night where he is in Australia. But again, he will be with us tomorrow morning to talk about his research in detail, which I'd urge you to watch tomorrow morning at 8:10. But I think we've got a little bit of video from Jordan. Steve.
Hi, I'm Jordan Hampton from the University of Melbourne. This is a quick thank you note to UFAW. I was fortunate enough to be awarded as one of the 2 joint winners of the Early Career Researcher Award in 2023. And it's an honour I'm really grateful for. I'd really like to acknowledge UFAW and also my colleagues that nominated me and provided letters of support. And it's really helped me to develop my career as an early career academic and also to build an international profile. And it's a nice opportunity to reflect on the beginnings of my career in animal welfare science, which started around 10 years ago when I almost fell into this field when working with feral camels in the Australian desert. And since then, I've really enjoyed investigating the welfare of more and more wild species, and it's a field that I think has a lot of growth yet to come. So I encourage any other early career researchers to apply for the award. I think it's an excellent initiative, and I'm deeply grateful for the help and support that's been provided to me. Thank you again.
Fantastic. So congratulations to Jordan when he wakes up in the morning. Like Jordan said, if you do want to apply or you want to nominate a colleague, please do consider following that link, ufaw.org.uk. ECR will launch the call for awards later in the year. So do nominate someone if you think they deserve next year's award. Moving on, my final job of the day is to announce the UFAW Medal for Outstanding Contribution to Animal Welfare Science, which this year goes to Professor Per Jensen from Linköping University in Sweden. Per's work will be known to many of you. He's had a real long-standing interest in the changes in behavior that animals undergo when they're domesticated. He's done lots of work in behavioral genetics, epigenetics, and all sorts of other fields, too numerous to mention, working with chickens, dogs, and many other species. He even has a colony of red jungle fowl to study in Sweden to compare with domesticated chickens. He's clearly an inspiration, not just for the many students and postdocs and so on that he's supervised and mentored, but also some of the leaders in the field who nominated him for this award and did describe him as an inspiration. So I think he doesn't need much more introduction than that. It's an incredibly impressive record of a long career, understanding some really fundamental things about animal welfare, but also using that information to influence and bring about change by activities like advising the EU on animal welfare policy and so on. So it's my absolute pleasure to award the UFAW Medal for Outstanding Contribution to Animal Welfare Science to Professor Per Jensen. And we're going to go straight into his talk where he can tell us more about some of the work which won him this award. Thank you very much.
So first of all, let me extend my sincere gratitude and thanks to UFAW for this really flattering medal award that I have received, and I'm so grateful and proud of this. and also for this opportunity to talk to you in this meeting. And of course, I could use a meeting like this now, or talk like this now, to go over everything I've been doing over the last decades or so in my research. But I think it's much more interesting and fun to actually talk about stuff that we are right in the middle of right now, and the plans for the coming years. So I will share my screen with you here, and actually talk about play behavior in animals, which is one of my new favorite topics, and where we have started a research program which I think is really promising and interesting. So play behavior, I think everyone knows what we are talking about, but let's just start by having a look at some example here of play behavior. And I'm convinced that you all share with me these kind of positive emotions that it awakes in a person who is observing animals playing like this. You see some social play here where the animals are wrestling and actually behaving a little bit as if they are aggressive, but we are, I think, quite clear about the fact that this is not real aggression. And we see some object play here, some locomotory play. Now, these are things which everyone knows about, and yet, as you will see, is extremely complex when you start looking closer at it. So to start with, one of the more sort of obvious problems with studying play behavior is this. So let's have a look at some play behavior in another species. These are piglets playing around. And again, I think that even if you are not particularly familiar with pig behavior, you recognize this as play behavior. You get this automatic feeling that these animals are having a lot of fun. Now let's look at something very, very similar. So when you watch these piglets, you also see the same type of behaviors. The behavior patterns are actually very similar. But again, I don't think you need a lot of experience with pig behavior to realize that these guys are not having fun. This is serious matter. This is aggressive behavior. And yet, very similar behavior patterns, very similar motor activities. In one context, this is having fun. In another context, it's a very stressful, aggressive behavior. So the first thing we need to do is actually to define what do we mean with play behavior. And there's been a lot of attempts over the years to offer some really solid, waterproof definitions. And I think that we are still basically there with these relatively old definitions that we see on this slide. So Beckhoff and Byers define play behavior as postnatal motor activity that appear to be purposeless. So the essential thing here is that motor patterns from other contexts are used in modified forms in temporal sequencing. I like to think of it as fractionated behavior, where you can see that there are kind of bits and pieces from different behavioral systems that are interspersed during the play behavior. Burkhardt has set up a number of criteria for what we should regard as playful behavior. So here is a summary of his criteria, which we still use in research. Play behavior does not contribute to immediate survival. It's not fully functional. That's basically the same as saying as it's fractionated. It's spontaneous, self-rewarding, and characterized by positive emotions. This is very central as something I will come back to. And also the last criterion, it occurs when the animal is free from stress. So when you start looking more carefully at the different types of play, I mentioned it briefly already on the first slide here, we can identify three main categories of plays: the locomotory rotational play, which the animals usually perform when they're on their own; the object play, which obviously involves manipulating different types of objects; and the social play that also involves other individuals. Now, obviously, there are no clear-cut boundaries here, and real play behavior often sort of mix these things together. So there may be locomotory rotational play in groups involving objects and involving some kind of mock aggression as well. This is from a review paper summarizing some of the different types of play behavior, but you can see some examples from different species, such as in pigs, you have locomotor rotational behavior, like this jump on spot, jump up and down, and so on. We have object play with an example from cats here, with pawing, patting, or biting inanimate objects. And we have social play with examples from rats, pouncing, play pinning. So play behavior. One of the really interesting aspects about play behavior is the fact that it's evolutionary, highly conserved. Looking at phylogenetic tree here, of part of the vertebrate group, we see a lot of vertebrates here, and these little squares shows in black groups in which play behavior, according to the criteria I just mentioned, have actually been observed and described. And we can see that this ranges from fish, ray fish, and also bonefish, to frogs and toads, turtles and lizards, birds, crocodiles, and of course all the way up to mammals. And what we see here on this picture is a lizard that is playing around with a dog toy. So what we need to understand here is that play behavior, because it's so evolutionary conserved, combined with the fact that it's a time-consuming and energetically demanding behavior, this basically suggests that this must serve some really important function, some evolutionary function. It's otherwise it's really hard to understand why this energetically and time-consuming behavior would have been maintained in so many different vertebrate groups. And in fact, also there are descriptions of play behavior in invertebrates. The other thing about play behavior is that it's particularly common in young animals. It's obviously not totally exclusive to young animals, because adults of many species, including humans, like to play, but it's more pronounced and most common in young animals. These are old studies from Jaak Panksepp, who followed the ontogeny of play behavior in rats in this case. And what we can see here on this graph is this typical peak during the early infancy of the young. And this goes for isolated rats that are allowed to play later on, and it goes for group housed rats. So it's basically the same ontogenetic development in these behaviors. So what about the function? What do we know about the function? As a matter of fact, not a lot. We know that it must somehow benefit the animal, otherwise it would most likely have been washed out by evolution a long time ago. So the common theory that I think most people would subscribe to, that it has something to do with practicing locomotional and social skills. So by playing, the animal gets to learn more about itself and its capacities, but also of its companions and its conspecifics. And here is, again, from a review paper, sort of an overview of how different types of play in cats, in this case, can contribute to functional development in different ways. So, for example, we have the mutual social play here, both with other cats and also with humans, that may be a way to learn about social manipulation. You have object play, which may be a way of learning about hunting and the physics of the world. The solitary play, which will increase the knowledge of self-image, coordination, and so on. This is all fine, and it looks very persuasive when you see a scheme like this. The big problem is that there is very little empirical support for anything of this. So people who have tried to follow up on this have looked at animals that, for example, have a lot of object play during their infancy. Are they actually better hunters when they grow up? It turns out they are not. animals that engage in a lot of social play when young, do they acquire social skills that they can use later in life? Well, research is not very convincing in this case. So I think that we still need to do a lot of research on this to actually know more about the function in the long run. One thing that we do know about play is that it's something that is closely related to animal welfare in a number of different ways. So these are examples from one study on piglets, showing that when piglets are allowed to grow up in enriched pens, not only do they later have less aggressive behavior, but they show more social play, more locomotor play, probably as a consequence of being in this enriched pen. So what we can say is that, yes, play, the opportunity to have to play may reflect some of their earlier welfare. This is also highly interesting when we look into the neurobiology of play behavior, which is of course highly relevant if we want to understand more about the welfare of animals in relation to the play behavior. We know, for example, that endogenous opioids, which are heavily involved and related to pleasure feelings of positive emotions, that these endogenous opioids are stimulated by play behavior. Oxytocin is stimulated by play behavior. So animals that play have a higher levels of oxytocin, and you can also stimulate play by providing oxytocin through, for example, nasal sprays to animals. And the same goes for dopamine, heavily involved in play behavior. This can be shown, for example, by the fact that dopamine receptor blockage will make animals cease playing, and the same goes for opioid receptor blocking. So yes, the whole reward system of the brain seems to be heavily tuned in with the capacity to play. So another part of play and neurobiology, which is quite relevant, is what's the relationship with cognitive capacities of the animal? And still, of course, we don't know much about this. There's a lot left to study about this. This is one example of an interesting observation that I think we can say so far we don't understand it very well, but there seems to be a relationship between brain size or total brain size and the frequency of play behavior. So this is kind of a meta-analysis paper that has gone through a whole bunch of mammals and measured their encephalization quotient, which is essentially just the size of the brain in relation to the size of the body, and what we know about play intensity in this species. And you can see this relationship: bigger brains, more play behavior. We don't know what this means. Is it causative, or is it just a coincidental correlation here? But there's a lot to be done here in relation to brain anatomy, brain physiology, neurobiology, and play behavior. So in my group, we are particularly interested, or have become particularly interested in one particular part of the brain, which I would claim has been largely overlooked over many decades of neurobiology work, and this is the cerebellum. And just to have a look at what goes on in the cerebellum, as a first glance into the cerebellum, if we look to typical mammals and in this case humans, we find that the cerebellum contains many more neurons than the cerebral cortex that we usually associate with cognitive processes and cognitive capacities and so on. This goes for many animals. So another aspect of play behavior and in relation to cerebellum is the fact that cerebellum is a part of the brain that develops a lot postnatally in mammals. So here we are back to the rats again, and we have the this line here, which is which shows you the play behavior ontogeny, just as we saw in a previous picture. And what we see here with the dotted line is the synaptogenesis in the cerebellum. So that's basically a measure of how synapses are developed and grow in the cerebellum. And you can see that there's a very close relationship. So during the period when animals play, or in this case, when rats play a lot, there is also a large proliferation of synapses in cerebellum. So this might be some kind of indication that the cerebellum is somehow important in relation to play and also to the later neural capacities of animals. So let's gradually move over to my favorite species here in this context. So obviously we work with chickens in my research group, to a large extent at least. We work with chickens, and we've done a great deal of work on things like domestication and domestication effects on behavior, brain development, and the interrelationship there, and also, of course, on the genetics of domestication and behavior changes. But in this case, let me just pinpoint a few things from some papers that we published a couple of years ago. So one of the defining aspects of domestication, commonly included in this sort of set of phenotypic changes that we usually refer to as the domestication syndrome, one of the central parts of this syndrome is that in basically all domesticated species, we get a decrease in brain size relative to body size. And here is a comparison we published some years ago, comparing body mass and brain mass in domesticated chickens and in their ancestors, the red jungle fowl. And of course, the domesticated chickens, usually, at least those that we look at here, laying hens, they grow much, much bigger. Their brains grow a little bigger, but of course not to the extent that it outcompetes the increase in body size. So the relative brain size actually decreases. And looking a bit more into details of this in different parts of the brain, we can see that this goes for basically all parts of the brain that we look at. So the cerebral hemispheres are larger in absolute measures in the domesticated chickens compared to the jungle fowl, which is the red lines here. Not much happens about the relative size. If we look at other parts like the optic tectum, again, the same thing when it comes to the absolute size, but in the relative sizes, the domesticated animals have a smaller optic tectum. They have a smaller brainstem relative to the rest of the brain. And then comes this amazing exception, and that's the cerebellum. So the cerebellum is, in fact, considerably larger in domesticated chickens relative to the rest of the brain. And this is the only part of the brain that is really not only defended evolutionary when it comes to relative mass, it's actually favored. So there is an increase in cerebellar mass. So the other interesting thing that comes from this graph comes from this first part of the graph here. So we have the age of the chickens down here at the bottom, and we can see here that the cerebellum is the only part of the chicken brain that grows in relative mass during the first few weeks of life. So it's a very plastic and in many ways an interesting part of the brain in this case. So the cerebellum is has a very special structure as well. It's heavily foliated, sort of increasing its surface space, and this is a way of packing in more neurons per volume brain. And again, we see that in the domesticated chickens, which is an example here to the right, there is more foliation, quite substantially more foliation. The graphs at the bottom here shows the foliation index of red jungle fowl and domesticated white leghorns. And we can see that this foliation index, which describes the degree of foliation of this cerebellum, is much higher in domesticated animals. So what can we say about the cerebellum? Well, the traditional view of cerebellum, if you look up cerebellum in a textbook that is maybe 10, 20 years old, you will see that it will probably state that this is the part of the brain that is involved in motor control. And that's it, full stop. And I think that's why people have not been very interested in the cerebellum over the past decades or so, because it's not thought to be a particularly interesting part of the brain. However, research in the last 20-ish years have shown that it's doing much more than that. For example, in primates, it has been shown to be involved in cognitive development and also very vital for social interactions and the understanding of social interaction. In rats and in chickens, from our own research, we have shown that it's quite involved in things like fear conditioning, memory consolidation. So the cerebellum is involved in many more cognitive functions that we have previously thought about. So it really deserves some appreciation and some interest. So yes, so our research that we are working on now is focusing on play in chickens. And what we want to understand more about here is the form that it takes. How do they play? What might possibly be the function of this play, both in a short time, approximate perspective, and in relation to neurobiology, and in the larger context of animal welfare. So this is a research program we have basically just started. It's just been going on for a year or two, and we have done we have some first publications on this, but we have many more plans and hopes over the next coming years. So I'll just give you a few examples from our research so far, and also about the plans that we have. So essentially, so far, what we have used as methods is that we have taken chickens out of their home pens. We have designed these so-called enriched playgrounds, which are basically just open space arenas with toys of different kinds that we think that chicks might like to play with. And we put them in this arena for various times. For example, in this case, 30 minutes, twice per week. We have videos that record what they are doing. And then, of course, we can analyze their behavior in there and quantify the behavior and put it in relation to different kind of things. Now, one common objection that I've had when I've been talking to people about this is that people are saying, what, chicks? Do they actually play? Never heard about playing chicks. And so the first thing we had to do actually was to have a look at that. Do they actually play? And in particular in this context of not being together with their parents, because as you all know, chicks in commercial settings, they never grow up together with their parents. So these are chicks that are placed on their own, of course, in these playgrounds. And I'll show you just a few examples from some footage that we have from one of our first experiments. So this first small video shows locomotor and rotational play in chicks. So you see and these are jungle fowl that we see now, the dark ones, but it's basically the same type of behavior. So we have the jumping, the running, the rotations, very much like you see in most mammals. I have an example here of some object play in the same way. So there are objects in here, little mealworms, little pieces of string and other stuff. And you see here how one chick picks up this object and basically runs away with it. And this elicits some social play as well, right? Because the other ones are following and trying to pick up this object and steal it from the first little chick here. And we see exactly the same thing here in the jungle fowl. Here's one chick that picked up this little object and examining it a little bit and running around with it. And this resembles very much, I hope that you see that, this resembles very much the dog puppies that I showed in my first slide here, that both run around with little objects and also engage in these running and hunting types of games. Then we have the social play, which in chickens mainly consists of this kind of play fighting behavior, very similar to what you saw in the piglets. And of course, this is you can mistake this for serious aggressive behavior, but in fact what you see here, and the reason for why we can say that this is play behavior, is because it's so fractionated. There are these outbursts of small aggression-like behaviors immediately followed by other types of behavior. So we quantify these different behaviors in red jungle fowl and in white leghorn domesticated chickens. At what we found was a lot of play. And we also found the same type of ontogenetic development that we have seen in the slides I showed you from the rats, for example, and we've seen in other species as well. Now, these are the different types of play that we see here. But we can just concentrate on the upper part here. This is the total play, so all the different types of play added up together. And what we see is a considerable increase among chickens that are domesticated. And this is also pretty much what we are expecting, because we know that as part of the domestication syndrome, we also have this thing which is called neoteny, or pedomorphy. So animals tend to be more juvenile, more childish as they move on to domestication. So we've also looked at play behavior in relation to how it's affected by early stress, and in this context we have looked specifically at chicks that come from a commercial hatchery, which contains a lot of very stressful handling procedure. And what we see is that, yes, they do also play in the same way. And in fact, when given the opportunity, these early stressed chicks tend to play a bit more than control chicks that do not have this experience of early stress. We don't know exactly what this means at the moment, but we think that it might be kind of a rebound effect, a dammed up motivation to express this, this type of play behavior. So to summarize bits and pieces of our own research, we can say that play behavior in chickens, they seem to be well conserved throughout domestication, but domesticated chickens actually play much more. We can also see that early stress alters some of the ontogeny of the play behavior, and we think that cerebellum size might possibly be involved in the function of the play behavior. So what we want to find out here in the future now is what is the function of cerebellum in play ontogeny? We want to find more about the neural substrates, but also we want to know if we can possibly stimulate chickens to play more and tax in on this very positive welfare effect that we know that that chicks have. That play behavior actually has, and we want to know if this can possibly improve their long-time welfare. So the program we are working on right now has to do with looking at connections between cerebellum size, structure, and also its genetic architecture, and later cognition and welfare. And we also want to look at the possibility to buffer early stress by stimulating chickens to play and see how this affects their cognition and their welfare. We have some pilot studies that have just been finished. So this is just a quick pull out of these pilot studies where we have had chickens from such a commercial hatchery, where we have provided them with extra stimulation of play during their first 5 weeks of life. And then we have looked at their behavior in different contexts. And what we see here is from a novel object test showing that chicks that have been stimulated by more play, they actually are more active in this special test, which is a sign of less stress, and they also interact more with this novel object, which is a sign of reduced fear. So even if these are really preliminary and early results, we are very hopeful that this might actually be one way forward. And with this, I'd like to thank you once again for listening to this talk, and also once again thanks a lot to UFAW for this extremely great honor of awarding me this medal. Thank you.
Thank you very much. And Per, I see that you are already with us to answer questions that people have asked.
Indeed.
Sorry, the technology is not cooperating. I'm just trying to pull up the questions.
So you need to pull out the question box, Lisa, so to sort of stretch it out. So where it says questions, you want to undock it and increase it in size.
I had done that, and
yeah.
I've lost that window.
Right. Okay. So you might. Can you got it?
I've got it. Yep. Thank you. Yeah, thank you very much for a really, really interesting, fascinating talk, Per. And we have already quite a number of questions that have come in, so I'll ask some of them if I may. One question is, how does cerebellum size and development for the domestic mammals compare to that of the chicken?
The answer is that I don't know, because I don't think that anyone has actually done this type of comparison. It would be really interesting, but as I said, I'm not sure. I mean, the hypothesis would be that if it does play some important role in coping with the domesticated situation, which means, of course, living close to humans and having a less fear, then we would expect it to have the same pattern. But as far as I know, no one has really done that comparison. So yeah, please do it, someone.
Fair enough. Sounds like a research area needing further work. The next question we have is how best can we consider play as a welfare outcome measure? Is it better seen as a short-term measure that is reflecting current positive states, or can its occurrence be used as a cumulative outcome measure considering the results you've shown with the relationship with early stress?
That's a really good question. At the moment, I think that for the best of what we know, I think that it's primarily something that has to do with the current state of the animal. So during play behavior, we have all these positive emotions, which can be reduced by negative experiences and by stress and so on. But I also think that some of the results that we have seen, for example, the very last slides that I showed you, indicates that there may be long-term positive welfare effects of play behavior, and I think that's pretty much what I hope to see in the future. So yeah, it's part of our research program right now, is to do this long-term follow-up and actually check and see, does this have any long-term effects or not? Definitely, it seems to be something that is important to the animals during their juvenile period, and as such, of course, it has its own right as a welfare measure. But I have this suspicion that it may be more than that.
Thank you. So one of the many next questions we've got is a comment on a question. Hi there, fully agree with the benefits of play. In my field of large animals, my experience to date, however, has been that the desire to play in monogastric animals, such as pigs and poultry, is transient, and they quickly become bored. Have you considered studying why this happens, or have you any suggestions how this effect can be overcome in order to maximize the benefits of play?
Well, I'm not sure that's my experience, actually, that the animals are getting bored. My experience is that, and I think that's sort of supported by quite a lot of research, that this is a self-rewarding activity, which means that in particularly domesticated animals seem not to be able to get enough of it. And I think that anyone who owns a dog knows that the urge to play, the motivation to play, is so strong and so rewarding that you can basically exchange normal food rewards during training for just a small bit of play with your dog. So the dog will do things to please you just in order to be able to get some tug of war or some chasing of a ball or something like that. And they don't seem to get tired of that at all. So I'm not sure I agree with the fact that they are getting bored. Having said that, what we are doing so far in our own research is that we have stimulated play by taking the animals out of their normal environment and putting them into this enriched play arena situation. And we really need to look more into what they are actually doing in their home environment. And I think most research so far indicates that you get an increase in play when you change the environment for an animal. So you increase the space, you add new individuals, you add toys or something like that. So maybe the correlation between what they do in this kind of stimulating situations are not exactly correlated to what they do in their normal home environment, but that needs to be looked into.
That's actually anticipating a question I had, which is how do we encourage play behavior, and to what effect does the immediate social environment and the composition affect their desire to play?
Yeah, I mean, it's quite easy to stimulate play, to get young animals going and play behavior, because it's basically to do something with their environment, to increase the space, to add objects. That will stimulate the play quite immediately. I actually think that that's from a welfare perspective, I think that we can stimulate play relatively straightforward. We have more of a methodological problem when it comes to the opposite. How can we stop animals from playing? Because in order to be able to do some proper research on this and see the consequences of play, we need control groups where we basically stop them from playing. It's not very fun to actually take that opportunity away from them, but from a scientific perspective, that is a problem. And so far, I haven't really come up with any good solution about how to stop animals from playing, because it's such a strong self-rewarding activity. So they'll basically do it, all the time.
Interesting. Could you please give us more explanation about your rebound effect hypothesis regarding the chicken experiencing early stress play more?
Yeah, well, I will emphasize that this is a hypothesis. I'm not even sure I would call it a hypothesis. Let's call it an informed guess more. So the idea here is, because we were pretty surprised actually. The expectation was that these animals that had a history of a pretty severe stress experience would actually show less play, because we were guessing, or we knew actually from other research that they already have a negative state of mind. They show a negative cognitive judgment bias. They have increased stress susceptibility and so on. So we know that they are heavily affected by this early stress. So we were expecting them to play less, and we found the opposite. They were actually playing more. So the idea was that, so this goes back to this correlation between what they do in their home pens and in these test arenas. So the rebound idea basically is a guess that maybe these animals did not play a lot in their home arena, and for that reason they have some kind of build up a motivation of play. So when we finally introduce them into this enriched large arena, basically they have this rebound effect that I was talking about. It's a pure guess, and I hope that we will be able to look more in detail into this, but so far this is the best explanation we can come up with.
Thank you very much. There are more questions, but unfortunately, I think we're probably going to have to stop here in the interest of time, but would very much encourage people to please engage after the session to ask follow-up questions. Thank you very much indeed for a fascinating talk, and many congratulations on a well-deserved award.
Thanks a lot.
So our next talk in the session is from Lindsey Kitchenham, who is going to speak to us about why lifelong conventional housing reduces the sociability of female mice. And I think that talk's just getting loaded up.
Hi everyone, I'm Lindsey, a PhD candidate at the University of Guelph, associated with the Campbell Center for the Study of Animal Welfare. And today I'm going to talk to you about why lifelong conventional laboratory housing reduces the sociability of female mice. Social interactions are important to mice. They are a social species and prefer to live with other mice rather than be alone, especially for females. They find social interactions rewarding and can have preferred social partners with whom they share greater amounts of time, proximity, resources, and affiliation. Having social companions can have benefits too, like buffering against negative affective states, and even promoting recovery from injury and disease. Thus, social interactions could have important consequences for mouse welfare. So how does conventional laboratory housing affect them? In conventional barren shoebox-sized laboratory cages, previous studies in our lab have found that female mice are more aggressive towards cage mates and engage in more harmful peer-directed behaviors like barbering. Compared to mice from well-resource enriched cages, conventionally housed mice are also less sociable and less attractive as social partners. Furthermore, there were signs that adverse home cage experiences mediated this behavior. Conventionally housed mice who received more aggression in their home cage avoided other mice from conventional housing and instead sought out companions from enriched housing. So our aims were to replicate these results from Adcock et al. And investigate further why housing affects sociability, social attractiveness, and agonism in female laboratory mice. Thus, we first asked whether aggressive or more aggressed mice are less sociable to see if we could replicate our previous findings. We also looked for a new effect, whether subjects who themselves are more aggressive are less sociable, since these mice could also be socially anxious, or maybe generally dislike or are disliked by other mice and become more socially isolated. In addition to agonism in the home cage, we tested two more hypotheses about housing-related mediators on female mouse sociability, attractiveness, and agonism towards strangers. The second is that conventionally housed mice may respond with depressive-like states, such as learned helplessness, pessimistic judgment bias, and depressive-like inactive but awake behavior. Here we hypothesized that much like depressed humans, social interactions could be unrewarding or even threatening. Third, conventional housing reliably induces stereotypic behaviors in mice, repetitive behaviors induced by frustration and/or brain dysfunction. We've previously found, too, that stereotypic mice become targets of aggression in their home cage. Maybe it's because stereotypic mice are behaviorally inflexible and so may struggle with the complexities and nuances of social interaction. Or they may be socially odd, aloof, much like humans with disorders typified by abnormal repetitive behaviors. So to test these hypotheses, we recorded home cage behaviors and ran a social interaction test. Our subjects were 15 black C57 mice who were socially housed their entire lives, either in conventional shoebox-sized laboratory cages, which were barren except for some nesting material, or large, well-resourced cages containing enrichments that are highly valued by the mice and validated to improve welfare. Our stimulus mice were pairs of black C57s or white balbs, one mouse from conventional and one from enriched housing, who were unfamiliar to the subjects. Home cage behaviors were recorded via live scan sampling under red lighting during the dark active period for mice. We recorded all instances of agonism given or received by cage mates, depression-like inactive but awake behavior, where the mouse sits still but is clearly not sleeping because she has her eyes open, and stereotypic behaviors where a mouse repeats an activity like route tracing or backflipping 3 or more times. These behaviors were recorded every 2 months over the 14 months lifespan of the mice, and then an average lifetime time budget was calculated. These behaviors were more frequent in the conventional laboratory cages. Sociability testing was also done during the mouse active period and took place in a novel neutral arena that we made out of corrugated plastic. First, the subjects were allowed 10 minutes to habituate to the empty arena and 4 empty stimulus cups, and she was then guided back to the center of the arena. Then, 2 identical novel objects and 2 stimulus mice were placed under the cups. One of the stimulus mice was from the conventional laboratory cage, and the other was from the enriched well-resource cage. For the social choice phase, subjects were allowed 5 minutes to explore while stimulus mice are trapped under cups. An observer blind to subject and stimulus mouse housing later scored the time subjects spent sniffing at the novel objects in each stimulus mouse, and we calculated subjects' preference for social stimuli with the sociability index as the ratio of time spent sniffing social stimuli over total time spent sniffing novel objects and stimulus mice. Subjects were then guided back towards the center again. Next, we removed the stimulus cups, allowing subjects to interact freely with their stimulus mice to start the 5-minute free social interaction phase. Again, we calculated the sociability index, and now we also counted the number of times subject mice initiated agonistic interactions with each stimulus mouse, as well as the number of times the subject mouse received agonism from each stimulus mouse. All data were then analyzed in SAS for academics using generalized linear models, and our results are as follows. First, we analyzed the effects of subject mouse housing, stimulus mouse strain, and their interactions on overall sociability of the subjects to see if we could replicate our previous results. We found that conventionally housed subjects were less sociable than enriched subjects, having less preference for social stimuli over novel objects in both the enclosed social choice and free interaction phases. Next, repeated measures models were used to analyze the effects of stimulus mouse housing, subject housing, and stimulus mouse strain and their interactions on their relative social attractiveness to the subject mice. During the enclosed social choice phase, conventionally housed bulb stimulus mice tended to be less preferred to their enriched counterparts to both enriched and conventionally housed subjects. But for C57 stimulus mice, housing did not affect their attractiveness. There were no effects on stimulus mouse attractiveness during the free social interaction phase. We also assessed whether conventionally housed subjects and stimulus mice are also more agonistic towards each other, using subject housing, stimulus mouse strain, and all their interactions. We also checked whether sniffing during the free phase was aggressively motivated by seeing if it positively covaried with subject displayed agonism. It did not. Neither subject nor stimulus mouse housing affected how agonistic they were during the free social interaction phase, but it did affect whether subjects were targets of aggression from bulb stimulus mice only, who were more agonistic towards conventionally housed subjects. Finally, to investigate possible explanation for these housing effects on subject sociability, stimulus mouse attractiveness, and agonistic interactions, we independently added the lifetime average of home cage time budgets for agonism given and received, as well as for inactive but awake and stereotypic behavior as covariates in the models. Home cage time budgets didn't predict housing effects on stimulus mouse attractiveness or agonistic interactions, likely because the sample size was small, since these effects were limited to bulb stimulus mice, but we did find interesting effects on sociability. First, we found that receiving agonism predicts low sociability. During the enclosed phase, there was a trending overall effect across both housing conditions, but in the free interaction phase, agonism received only predicted low sociability for subjects with C57 stimulus mice. We also found that subjects who themselves are agonistic were less sociable. This effect was significant across both housing conditions in both the enclosed and free interaction phases, but much stronger during the free interaction phase. We also found that high levels of inactive but awake behavior predicts low sociability for all subjects during the free interaction phase. During the enclosed phase, the same pattern was seen, but only for subjects with C57 stimulus mice. Finally, subject stereotypic behavior predicted low sociability only in the enclosed social choice phase, where it was driven by conventionally housed subjects and subjects with bulb stimulus mice. So in sum, conventionally housed subjects were less sociable than enriched subjects, which replicates our previous results. And welfare-relevant home cage behaviors seem to mediate this effect, but these new results now need replication with a bigger sample size. We also don't know why aggressive, picked on, depressed, and stereotypic mice are less sociable. For example, are conventionally housed mice anhedonic, no longer finding reward in social interactions? And how do these housing effects feed back on mouse welfare? Also, what cues are mice using to tell each other apart by housing? All are questions for future studies. But if you would like to know more, you can find our paper in Applied Animal Behavior Science. The authors acknowledge that this work was completed on the treaty lands and territory of the Mississaugas of the Credit. We'd like to thank NSERC for funding this research and provincial scholarships for supporting us students, the staff at the Central Animal Facility and Michelle Chiplack, our technician, and of course the mice who participated and the people who adopted them to good homes. Thank you for listening. I look forward to hearing your questions.
That very interesting talk. I would like to invite you to, if you're available, Lindsey, to please join us for some questions that we have.
Hi, can you hear me?
Hi there. Yes, we can. Thank you very much.
Okay, thank you. I apologize for the computer fan in the background. If I need to speak up, please let me know.
No worries at all. Thank you. So do you think that if females are housed together in different stages of, I guess, at different ages, could that influence their behavior? That if they're younger, is behavior better when they're housed together? I think I'm understanding that. I hope I'm not mangling that question.
Yep, so in our previous study, the one by Adcock et al., we did look at this effect in younger mice. We still found the reduced sociability, but the correlates of that sociability were slightly different. So we think what's happening here is that the long-term effects of being housed in that conventional laboratory caging are accumulating over time and making things kind of worse and mice less sociable, more socially isolated. So certainly I would expect to see differences at different age stages. Yeah, because I think these are cumulative effects.
Thank you. The next question that we have is, are there other breed differences that may explain your findings that only sociability affects in the bulbs?
So I think they're asking perhaps about different strain differences, so strains that we've not looked at. So aside from the bulbs, or are they? I think maybe perhaps they're asking if there are different correlates that would explain the bulbs different. I can talk a little bit. Since I'm not sure how to answer this question, I can talk a little bit about what's known about the strain differences. So typically C57 mice are assumed to be more of a sociable strain than the bulbs. The bulbs are often used to model, for example, autism, because they're typically seen as being less sociable. But what's interesting about this work is we've shown sort of with the attractiveness of the bulb mice kind of being enhanced in the enriched housing, is that might have to do with the type of housing system that they're kept in. So these sort of strain-based differences are interesting because they tend to interact with environmental conditions. So they're more genotype environment interactions.
Yeah, thank you. Yeah, I guess one of the, you already talked about some of the follow-on work that you guys are wanting to investigate, but I guess just from what you've done already, are there some implications or some recommendations that you might already have developed as a result of the work that you've done and the findings that you've had?
Yeah, so definitely in terms of laboratory mouse housing conditions, it's suggested in Canada, and I would assume many places elsewhere, to house mice as social, in social groups, because they are a social species, especially for female mice. Now, one thing that happens over here is when you have male mice, typically those individuals are housed on their own, and even more so if you want to enrich their housing, because you're worried about resource guarding effects and artificially enhancing aggression between the male mice. So these results might indicate that actually the enriched housing, despite the worry about enhanced aggression due to resource guarding, might actually help the male mice be more sociable as well. So that's another future avenue that would be directly applicable to changing current practices and policies in laboratory units.
Thank you. We have another question, and again, I'm going to have to see if I can try and intuit what was intended in the question. Is there a way, I think is there some manipulation that could be done to increase mouse sociability?
To increase it perhaps in the conventional housing conditions. Well, our results suggest that the decreased sociability is a direct effect of poor welfare. So I would assume that any kind of interventions that you would implement in that housing condition that increase welfare, so perhaps more gentle handling or less invasive laboratory investigation type studies, those would also help to improve sociability.
Thank you. So you talked about a number of things that you're sort of thinking about that need to be investigated further. Do you have particular next steps that you've got planned at the moment?
Certainly. So, yep, next steps would definitely be investigating similar effects in male mice that are in enriched housing, and we're piloting some studies on doing that safely. Potential avenues for how mice are telling each other apart based on housing involve analyzing different behavioral posturing, different urinary protein profiles that may be different. So are they smelling the difference between the individuals, or are they seeing the differences in behavior, or perhaps are these mice emitting ultrasonic vocalizations that might be indicating that they're from different types of housing?
Fascinating, thank you. So if it seems very likely that having an enriched housing is really improving sociability and ultimately, of course, improving welfare outcomes, what's the balance that can be made in terms of pragmatic recommendations for changing policies and guidelines? But obviously, you know, there are limitations in terms of how extensive an enriched environment can be provided.
Yeah, exactly. And I would think that depends on what baseline an institution is starting with. So we can start with perhaps the University of Guelph in Canada, where I come from. Our baseline is basically a shoebox sized cage with some bedding material, perhaps a cup for shelter, and some nesting material. And so at each stage that you add enrichments or improve the housing, right, they're little increments. They have incremental effects on welfare. So I think really being pragmatic, it would depend on where you're starting with. Institutions that are starting with even worse housing, so say no nesting material or no cups or even isolated housing. Now that would be a big jump. You would likely see a huge improvement if you were to even just put a little bit of welfare enhancing materials into their cages.
Fair enough. Great. Thank you so much. In the interest of time, we're going to have to leave it there. But thank you so much for a really interesting talk. And for anybody who would like to send any follow-up questions, please, by all means, use the Slack channel for the conference. So with that, thank you, Lindsey. I will now introduce Cynthia Schuck-Paim, who's going to talk about the topic of animal welfare science must look at time.
Good afternoon, everyone. My name is Cynthia Schuck. It's my pleasure to be discussing this topic with you on why animal welfare science must look at time. So, as we all know, time is at the heart of how any living creature experiences life. Affective states, whether positive or negative, will matter more or less depending on their intensity, but also critically on their duration. So it's not the same thing to be in a state of hunger just before a meal, or for weeks, months, or a lifetime. And the same goes for any other affective experience that an animal may have, such as cold, fear, hunger, boredom, the pain of a fracture, frustration from the deprivation of motivated behaviors, and so on. So to give you an example that is closer to us, we can see here the description of the pain of a bone fracture as narrated by someone who has gone through this experience. And we can see that the impact of this pain on the patient is very much a function of the intensity of the pain in the vertical axis and the duration of the pain on the horizontal axis. So there's very intense pain immediately in the hour after the fracture, then somewhat less intense pain during the inflammation period, but still it's painful. Then the pain gradually subsides as the callus starts forming and the bone is remodeled. Now, the same logic also applies to animals. We have here the case of an egg-laying hen that has just suffered a keel bone fracture, and the impact of this fracture on the welfare of the hen is also a function of the intensity of the pain and the duration of the pain. So we can expect a very similar pattern to what we observed in the case of the human patient. We could also have another example, for example, a hen that suffered an acute fatal episode of egg peritonitis that got infected and the hen dies from septic shock. So in this case, there is some discomfort in the beginning before infection sets in. Then pain gets very, very high during sepsis. It's a very painful condition and then subsides abruptly as the hen gets into septic shock and dies. So obviously affective states are modulated by a number of factors, but intensity and time are key factors. So we can think of what would be the worst suffering in the world. And one good answer to this would be that with the longest possible duration and the highest possible intensity. Now, time-based metrics of well-being haven't been used much in the animal welfare sciences, but they have been used a lot in other areas such as global health from where we come from. One of them, for example, is the DALY, Disability Adjusted Life Years, which measures the time of healthy life lost to disease or to premature death. So, for example, here we have the case of a person from birth to death which experiences in the red squares here some episodes of disease, and in the last bout the person dies at 60 years of age, so 10 years before the average life expectancy for this demographic group. And the DALY here in this case is the measure of the years lost to disease weighted by a disability weight, which how much the disabled made the person unhealthy. So 5 years here in a diseased state with a disability weight of 50% would mean 2.5 years lost. This is calculated for all the disease episodes over the life of an average person with this disease. And the DALI here is simply the sum of the years lost to disease, so 2.5, 1.5, and 3.5, which is 7.5, and the years lost to premature death due to the disease. So DALI is 17.5 years. And by having this universal metric, which is time, you can compare very different conditions, injuries, diseases, any problems you want to look at in terms of the time they cause the person to lose to the disease you're analyzing. You can compare different age groups, different locations, different, demographics, evolution of the burden of the diseases over time, and set priorities for interventions, for funding, for research, etc. So with that in mind, we understand that in the animal welfare sciences, a biologically meaningful metric of welfare, of suffering, that is amenable to comparisons, a universal metric such as a time-based measure, is needed to quantify and compare the impact of very different welfare problems, policies, living conditions, to enable integrating animal welfare quantitatively into social, environmental, and economic analysis, and to raise awareness about how much suffering is embedded in each unit of animal products, in each kilo, in each liter, in each egg, with a metric that anyone can understand, that is not abstract. So based on our experience in global health, we developed the cumulative pain framework, which measures the time in unpleasant, negative, affective states. And we use the term pain as a shorthand for any felt negative affective state. So time in pain of different intensities enables comparing very different outcomes, very different hazards through their effects in terms of the time the animal spends in pain, or the average member of a population spends in pain. For intensity, we use 4 discrete categories of pain intensity, from annoying, which is simply a discomfort, to excruciating. These categories are defined by tight criteria. Don't worry, I will not read this for you, but mostly the level of disruption in terms of behavior. This is one criteria. The expected effect of drugs, painkillers, psychotropics on behavior, effects in terms of attention, cognitive performance, and so on. So you can read the definitions in the website if you're interested. And we have a notation system which we call pain track, where we make hypotheses about the duration of the pain associated with a welfare challenge and the intensity of the pain explicit in this notation system. So for example, here we have a pain track for the case of a Q-bone fracture in laying hens. And the probability that the pain belongs to each of these categories of pain intensity is informed by multiple indicators. So behavioral indicators, the effect of drugs on pain and behavior and physiological markers, physiology, immunology, knowledge of the neurological system associated with the structure that was affected, tests of preference, of motivation, evolutionary reasoning on the adaptive importance of the pain of that negative affective state associated with the hazard, evidence from closely related species in terms of pain, and so on. So we have this hypothesis here with uncertainty intervals, and the better the evidence, the narrower our interval of uncertainty, both for duration and for the probability that pain belongs to each of these categories of intensity. And once we have the hypothesis for intensity and duration, they are converted to time in pain by simply multiplying this probability here by the duration of the segment. And once you add up all the time in pain of the different intensities. You have the cumulative time in pain due to a fracture in this case. We have applied this framework to answer a number of questions regarding the welfare of egg-laying hens. The results are available in this book. And questions such as, would it be possible that due to the greater frequency of challenges such as aggression, fractures, infectious diseases in cage-free operations, welfare of the hens would be worse as compared to caged operations? Or even if welfare is better in cage-free environments, how much better is it? In the case of broilers, we ask questions such as how much suffering can be averted by setting genetic limits to the growth rate of broiler chickens, or would a longer life in the case of slower growing breeds, under conditions that are still better but far from ideal, lead to more suffering because the chickens live a longer life? And to answer these questions, we investigated a number of welfare challenges that laying hens and broiler chickens go through: lameness, behavior deprivation, fractures, injuries, thermal stress, cardiovascular conditions, infectious diseases, and so on. And in doing so, and because we had to look at both indicators of intensity of the pain and duration of the pain, it became very clear to us that very little attention in the welfare sciences has been paid to understanding the duration of the welfare challenges that the animals undergo, so how long they last, the timing of onset of these challenges, so when they emerge, and the evolution or clinical evolution of these challenges, so how quickly they progress and how they progress. There's very, very little information for many of the welfare challenges that are commonly investigated in the welfare sciences. So in this table we have some examples. For example, regarding egg peritonitis, which is both a chronic condition in survivors and a fatal condition, is the most important production disease in poultry. There's very little to no knowledge on the duration of the pain associated with it, on the duration of chronic pain in survivals, how quickly it progresses, how long the hen affected by it spends in critical stages. In terms of sepsis, this is the main cause of carcass condemnation in broiler chickens, and yet there is very little information on the clinical evolution of sepsis, even though this is an extremely painful condition, but it's a neglected condition in the animal welfare sciences. We don't know the time it takes for the hen to die from sepsis when loss of consciousness happens. In terms of Q-bone fractures in laying hens, the duration of pain, the clinical evolution of pain, inflammatory case, callus formation, and etc. We don't know much about it. In terms of injurious packing, fatal cannibalistic attacks, vent packing bouts, we don't know, in the case, for example, of a fatal cannibalistic attack, how long it takes for the victim to die. So what is the typical duration of these attacks? We don't know. The frequency of the bouts, the duration of the bouts, is it like a few seconds, a few minutes, many minutes? In terms of gait impairing conditions, lameness, when does it happen? I know it's a multifactorial condition, but we don't know much about the duration of each of the phases of lameness, how long each broiler, for example, spends in each gait score before it progresses to the next one. The same goes for cardiac abnormalities, the main cause of death, so sudden death, ascites, and the expression of motivated behaviors. What are the changes in the time budget over the production cycle as the hen gets old, as the hen or the broiler gets older? So there are many research gaps associated with the time spent in different conditions, when conditions emerge, how long they last, how quickly they progress. And these gaps are not only present in terms of poultry research, but also in other areas of animal welfare research, research on pigs, fish, etc. And it's very, very critical to understand these questions related to time, to onset, to duration. I'll give you an example. For example, we know that when we have less risk factors for a disease, let's say for cardiovascular diseases, for diabetes, etc., the onset of the disease is likely later on in life. So in the case of production animals, farm animals, we have the later onset of chronic disease and injuries relative to slaughter age when you have better genetics. For example, when you have a slower growing breed. And with the later onset of disease relative to the slaughter age, you're going to have a shorter time spent in pain until slaughter in the case of chronic conditions, because the disease emerged much later in life. You're going to have less time for disease progression. So what this means is that the disease will probably be less severe. It will not reach the most severe stages. And as a consequence, less individuals, because it takes more time for the disease to emerge, less individuals will develop the problem. So you have reduced duration, reduced intensity, less severe disease, and reduced prevalence. And as a consequence, reduced suffering. So understanding the effect of genetics, for example, or environmental conditions on the onset, on the timing of onset of disease is critical in this sense. And although the idea is not to present the results of the research conducted with this method, some very interesting findings emerge when we look at the effects of, when we consider the duration of the welfare challenges in animal welfare analysis. So, for example, our estimates show that chronic hunger in fast-growing female breeders, broiler breeders, is possibly the greatest source of physical pain for any farm chicken, including egg-laying hens and broiler chickens. And the case sepsis, for example, and the diseases leading to sepsis, such as egg peritonitis in breeders and laying hens, is possibly the greatest source of excruciating pain in these animals. So many interesting things emerge when we take time, duration into account. So thank you so much for taking the time to watch this talk, and we're available for questions.
Excellent. Sorry about that. I'll try third time's the charm. Cynthia, thank you very much for that very interesting and informative talk on a really important topic that probably we still really need to learn a lot more about. Time is a little bit short, but we've got a number of questions already up in the question window, so I'll just start working through them if I may. Do you think that pain itself could influence perception of the passage of time? That is, that severe pain could be perceived to have a longer duration than less severe pain? And is this ever the case in human conditions?
Yeah, definitely. As based on what we have in reports from humans and research in humans, then that could be certainly a possibility. Right now we are looking like at a broader picture, but these modulators of pain and of the perception of the passage of time are certainly there. There's some indications that by the end of life, even in animals, you may have a different perception of the passage of time, regardless of whether you are in pain or not. So it is a possibility. I'm not aware of research looking at that, but based on what we see on humans, definitely.
One of the questions I had actually is, do you think that some of the paucity of literature on pain in chickens is at least in part a reflection of our inability to accurately perceive or evaluate pain in chickens? And to what extent is that a potential barrier to research? And do you have any suggestions about how to better detect pain?
So could you just repeat the beginning of the question that was broken, Lisa?
Oh, sorry. Is it possible, is the fact that there isn't that much literature at the moment on this pain perception in chickens in part a reflection of our inability ourselves to perceive or evaluate pain accurately in chickens?
I think it's both. It's also, of course, difficult to understand pain and suffering in non-verbal creatures, and that's what welfare science is trying to do, investigating markers and behaviors, etc. So there's part of the question, but also I think it's perhaps the historical trajectory that the area is taking. I think there could be more research focused on some individual tracking of animals, so longitudinal research on the welfare of the animals, individual-based research instead of population. Of course, it's difficult because you have farms, and but now with precision, technology, it could be easier to track individual animals and understand what happens in terms of behavior, and combine them, let's say, with necropsy studies that take samples from specific periods in times from apparently healthy individuals. So there could be an increased emphasis on things that matter for the welfare of the animals at the individual level. So some of these things that I've mentioned, and of course there are others, but like how long it takes for the individual to heal from disease and injury, a trauma, etc. Or when does it happen, how quickly it happens. So I think there could be more studies in this direction.
Yeah, thank you. And I guess following up on an earlier question about perception of time, there's a question, should we or how could we try to take into account the possibility that animals may perceive time differently to humans and may or may not be able to perceive time or predict the end of an event?
Yes, that's another possibility. We may have interspecific differences in the perception of time. So this is not much of a problem when comparison is within species. So like in the case I mentioned with the broilers. So what is more painful, the chronic pain, the chronic condition of a hungry state for a long time in broiler breeders, or the confinement in cage hens, or what is our priority in terms of welfare challenge for chickens in general. So you are still within the intraspecific level of comparison. So you may not expect any large differences in the perception of time. When you're comparing a chicken with a cow, with a human, with a dog, then that might be a problem. And that's the general problem of interspecific comparisons, because you may have differences in perception of time, or in the capacity for welfare, or the intensity, the range of intensity that is perceived may be narrower, may be larger for different species. Some may be more stoic, others may be less, depending on whether they are prey or predators. And these are all open questions in addition to the ones I mentioned. So we don't know much. There has been some research on interspecific differences, but more on the philosophical level than on the welfare level, I think. So this is all open to investigation.
Yeah, fair enough. Thank you. We have run a little bit over, and apologies to all of our attendees, but it's the end of the day, so hopefully people don't mind giving us a couple of extra minutes. Thank you very much indeed, Cynthia. And again, if anybody wants to follow up with further questions, please do use the Slack.
Thank you so much.
So with that, that brings this session to an end, and I wish you all a good evening, and I'll hand off to our organizers in case they have any parting words for the evening.
No, I don't think so. Thank you very much for everyone who's chairing. Oh, here we go.
No, I don't have any parting words other than see you at 8 o'clock tomorrow morning if you're coming back tomorrow. Remember that we start a bit earlier tomorrow, but other than that, thank you to Lisa, and thanks to everyone for coming along today, and we look forward to another exciting day tomorrow.